Michel Sadelain, Herbert and Florence Irving, Professor of Medicine, Director of the Columbia Initiative in Cell Engineering and Therapy, Columbia University, United States. BBVA Foundation Frontiers of Knowledge Award in Biology and Biomedicine. Could you explain what CAR T cells are and how your basic research revealed their therapeutic potential to treat cancer?
We are fortunate to, all of us, have a wonderful immune system that protects us from invaders like viruses or parasites or bacteria. But when it comes to cancer, our immune system is not always ready to take on that challenge. Many years ago, a few of us thought that perhaps we should help this immune system of ours by engineering it, by teaching very important cells of the immune system that we call T cells to recognize cancer cells and then to proceed to kill and eliminate. And that's how we created these molecules that I named CARs, or chimeric antigen receptors. When we introduce a CAR into a T cell, that T cell acquires the ability to recognize tumor cells and then will proceed to eliminate those cells. We first tested that in animal models after about 10 years of research, and when we optimized it, we embarked on clinical studies which we had to conduct in the academic setting in our hospital. And here we are today with the first CAR T cell therapies approved by the US FDA for the treatment of some cancers, such as leukemias or lymphomas or multiple myeloma.
How did you first become interested in this field, and what motivated you to carry out this research?
I first went to medical school in Paris, France. I loved medicine and the contact of patients, but I soon realized that I was most excited by trying to understand the pathology underlying different diseases. And so I embarked on a PhD, which I pursued in immunology in Canada. And it is in the course of those studies that I started imagining that we should not just rely on the natural immune system, but help it by engineering it to acquire properties that go beyond what nature has endowed the immune system with.
At the level of clinical applications, what has been demonstrated so far about the potential of these therapies to treat blood cancer, and how many patients have already benefited from them?
The first clinical studies testing CAR T cells in cancer patients focused on leukemias and lymphomas. And that is because we had identified a target called CD19, a molecule that exists in many of these leukemias and lymphomas. Our animal models were based on the targeting of CD19. And so the first clinical trials started in patients with relapsed refractory leukemias and lymphomas, meaning that they already had undergone what's called the standard of care, and their cancer nonetheless was progressing. Very soon, the first clinical results reported by us, by Carl, and a few others later confirmed that these CAR T cells could be efficacious in these blood-borne cancers. The next cancer to be effectively treated by CAR T cells is called multiple myeloma. That is another blood-borne cancer. And today it is widely accepted that engineered immunity, exemplified by CAR T cells, can succeed where no other treatment, chemotherapy or bone marrow transplantation, has succeeded before.
To what extent has it been shown that CAR T cell therapies could also be effective against solid tumours? And when do you foresee that this could benefit patients who suffer from lung, breast, colon or brain cancer?
The first cancers to be successfully treated using CAR T cells were leukemias and lymphomas. And that's because we had identified a target called CD19, against which we could direct those CAR T cells. Thanks to the story of CD19 CAR T cells, their successes in the clinic, today the scientific, medical, pharmaceutical establishments are convinced of the value of engineered immunity. And now it is our job to make this work for many other cancers. If you apply the same recipe, if I can say so, for the treatment of solid tumors, the CAR T cells don't work as well as they do in leukemia and lymphoma. We are today starting to understand the additional barriers that need to be overcome in the context of solid tumors. Those include the selection of targets and the composition of these cancers, which are different from leukemias and lymphomas. I believe there's every reason to be optimistic that in the next years or certainly decade, we will see effective CAR T cell therapies for lung cancer or brain tumors and others.
What has been demonstrated up to now about the potential of these therapies for infectious diseases and autoimmune diseases?
When we identified CD19 as a potential target for future CAR therapies, we knew that a side effect of those CAR T cells would be to eliminate not only leukemias or lymphomas, but also normal B cells. These are the cells that produce antibodies, and antibodies, by and large, protect us against infections and other diseases. But sometimes antibodies have a bad effect. They attack our own tissues and cause forms of disease that we call autoimmune diseases. Investigators recently tested the CD19 CAR T cells that had been developed for cancer in patients with refractory autoimmune diseases, starting with systemic lupus erythematosus. Remarkably, marvelously, they immediately achieved complete responses in those patients. And this has started today a wave of new clinical studies in the worlds of rheumatology and neurology to try to control autoimmune diseases using CAR T cells. CAR T cells may also prove to be of benefit in some refractory infectious diseases. Think, for example, at the reservoirs of HIV, which no drug today can eliminate. We hope that perhaps CAR T cells may be able to succeed where none of the drug therapies have so far succeeded.